My name is Rob Willems, Professor of Population Genetics of Antibiotic Resistance in the Department of Medical Microbiology of the University Medical Center Utrecht in the Netherlands. And with this video, I'd like to introduce you to the JPI AMR funded project STARCS. STARCS stands for Selection and Transmission of Antibiotic Resistance in Complex Systems. The aim of STARCS is to characterize and quantify the processes of selection and transmission of AMR genes and antibiotic resistance within and between ecosystems, and to integrate these elements into predictive models that we'll use to inform policy development. The consortium consists of 8 partners from 6 countries. Of STARCS consists of 3 work packages. In work package 1 we have developed innovative methods to study AMR genes in complex systems. These methods will be used to study the selection and transmission of AMR genes in different ecosystems, like experimental animals in mice and dogs, in households between humans and dogs, and in patients. The results of these studies will be used to build epidemiological modeling approaches to characterize and quantify the dissemination of resistance. In work package 1, we developed innovative methods to recognize and reconstruct plasmids in bacterial genomes. For this, we developed MLplasmids and GPLAS, 2 new algorithms to reconstruct plasmids from short-read sequence data in order to determine the role of plasmids in host adaptation and to study the epidemiology of antibiotic resistance plasmids. In order to detect AMR genes in metagenomes, we further developed and validated ResCap, the first targeted metagenomic platform to analyze resistomes with a 200-fold higher sensitivity and specificity than metagenomic sequencing, and improved the bioinformatic tools to analyze the data. To determine the genetic context of AMR genes in metagenomes, being a mobile genetic element or a bacterial host, we further optimized Meta3C, which is a proximity ligation approach that binds context in a sequence-independent way by quantifying and exploiting their 3-dimensional collision frequencies in vivo. So it allows to reconstruct genomes as well as to assign plasmids and phages to the host in metagenomes. Finally, we optimized metatranscriptomics to detect and quantify the expressed AMR genes in complex samples by depleting eukaryotic and ribosomal RNA, and succeeded a 600-fold enrichment for resistant gene transcripts by combining it with ResCap. In work package 2, we used the methods developed in work package 1 in several datasets. One dataset will come from a Peking duck model for selection and transmission of resistant E. coli. Due to several reasons, including lockdown of laboratory facilities due to corona, this experiment was delayed. Luckily, we were able to finish the experiment just before the corona lockdown, but we expect to do the analysis of the collected samples later this and early next year using the methods developed in work package 1. In this experiment, 15 Peking ducks were inoculated with E. coli strains that are resistant against enrofloxacin, a fluoroquinolone, but also a cephalosporin-resistant E. coli strain and a susceptible E. coli strain. We used them in different combinations and in different concentrations. The antibiotics were also added to the water of the pond. Feedset samples were collected from different time points and will be used to assess colonization levels, transmission and persistence of resistant bacteria in the ducks using the tools developed in work package 1, such as ResCap, Meta3C, and metatranscriptomics. In another in vivo model using mallard ducks, we showed that HBL E. coli strains used to inoculate the birds, these are the colored bars in the figure, were able to colonize these birds during prolonged period of times in the absence of antibiotic selection, but also that these strains were able to rapidly spread between the birds. In a second experiment, mallard ducks were inoculated with a mix of different quinolone-resistant and susceptible E. coli bacteria, and ciprofloxacin was added to the pond at different concentrations to determine the minimal selective concentration, the MSC. This revealed different MSCs for different mutants, with one GER-A mutant indicated with a red arrow, outcompeting the susceptible strain even in the absence of antibiotics. 3 other studies investigated the transmission and selection of resistant bacteria in hospitals and between farm animals and humans. In this study, we looked at selection and transmission dynamics of ESBL-producing Enterobacteriaceae, ESBL-E in the figure, particularly Escherichia coli and Klebsiella pneumoniae, in hospitals, wards and rooms under the effects of different infection control strategies, being standard precaution SP and contact isolation CI. First, we identified pairs of potential ESBE donors, patients admitted with an ESBL-E, and acceptors, ESBL-E negative patients on admission, patients that were positive at admission but turned negative and positive again, and patients that were positive on admission and remained positive but with a different species. Subsequently, we assessed whether acquisition was due to clonal transmission or horizontal gene transfer of resistance plasmids. Plasmid similarities between donor and acceptor were based on ink typing and a plasmid-based MLST scheme. We identified in 21 pairs clonal transmission in both standard precaution as contact isolation involving both E. coli and Klebsiella pneumoniae, and 18 pairs of potential horizontal gene transfer with 8 pairs in which we identified interspecies transmission. In the same study, we also identified persistent circulation of a multi-drug resistant Klebsiella pneumoniae ST48 clone with multiple chromosomally integrated copies of BLA CTXM15 during the study period, which was between 2014 and 2016. The first and the last strains were isolated 535 days apart from rectal screenings of patients, and these showed a maximum of 16 core genome SNP differences. This clone carried 2 to 5 copies of BLA CTXM15 in its chromosome. In a next study, we investigated mode and dynamics of VAN-A type vancomycin resistance dissemination in Dutch hospitals. For this, we compared clones and plasmids based on combinations of short and long read sequencing and the new plasmid reconstruction tools MLPlasmids and GPlas developed in Work Package 1. We estimated the contribution of clonal spread and horizontal transfer of VAN-A resistant plasmids and transposons into the dissemination of VAN-A type of vancomycin resistance depicted with the different color bars. The data showed that on average, clonal spread was the predominant mode of vancomycin resistant dissemination, followed by transposon mobilization and spread and horizontal transfer of plasmids. However, we also observed regional differences with spread of the VAN-A transposon being predominant in the province of Overijssel. In the last study in Work Package 2, we analyzed the distribution of sequences, assembled contigs, encoding AMR genes to see how they are distributed across human and livestock isolates in the Netherlands. This figure shows a core genome phylogeny of ESBL E. coli isolates. The source of the isolate is shown in the multicolored inner ring. The red lines on the outside indicate the presence of sequences in the assembled contigs that encode a BLA CTXM15 gene. The length of the line indicates the length of the contig sequence, and internal connections show where pairs of isolates share an identical contig carrying BLA CTXM15. From this analysis, it became clear that connections were not restricted by E. coli lineage, but by source type, humans and livestock. Furthermore, we also found that for other AMR genes, connections were found between humans and cattle isolates, but were absent between human and chicken isolates, indicating that transfer of resistance between animals and humans might be limited. A mathematical model was used to model the impact that antibiotic usage in livestock has on human foodborne illnesses. Livestock antibiotic usage was assumed to have 2 effects: increase clearance of potential human antibiotic-susceptible pathogens from livestock, and 2, modulate the conversion of antibiotic-sensitive bacteria to antibiotic-resistant bacteria in livestock. The results of the model in the top figure show that decreasing livestock usage of antibiotics, going to the left, results in increases in overall human foodborne diseases due to decreased rate of clearance of potential human pathogens from livestock, while increase in antibiotic usage in livestock going to the right will lead to less foodborne illnesses, but a much larger proportion of the foodborne illnesses will be antibiotic resistant. Ideally, we wish to curtail all usage of antibiotics in livestock, but as shown in this figure, this will result in more foodborne illnesses. The bottom figure shows that these increases can be mitigated by decreasing the transmission from animals to humans on the y-axis and or by decreasing the transmission from animals to animals on the x-axis, both in theory possibly through better hygiene or farm management. Mobile genetic elements like plasmids and phages are central in the adaptation of bacteria to the selective pressure of antibiotics, and AMR genes carried on these mobile genetic elements can swiftly disseminate within and between bacterial populations through horizontal gene transfer. Insight into the flow of these mobile genetic elements is pivotal to control the spread of antibiotic resistance. Tools to detect and reconstruct plasmids from short-read whole genome sequence data are becoming more and more available. Two of these bioinformatic tools were developed within the STARCH project, MLPlasmid and GPlas, and they have and will in the future importantly contribute to our knowledge on the quantitative role of plasmids in bacterial adaptation and the dissemination of antibiotic resistance. However, reliable detecting and reconstructing mobile genetic elements to follow the transmission chain of these elements along gene flow networks in metagenomes is still challenging. Therefore, further bioinformatic tools to detect and reconstruct mobile genetic elements from short-read metagenomic datasets are needed to inform us on the flow of AMR genes in and between complex ecosystems.